Welcome to the Development in Sea Urchins module. In this talk, we're going to learn about the fertilization and cleavage processes in the context of sea urchins. Sea urchins are marine, spiny, globular invertebrates. They are deuterostomes that belong to the class of Echinoidea that comes under the phylum of Echinodermata. Other organisms that also come under the phylum of Echinodermata are starfish. Sea urchins have been used as model organisms to study different developmental aspects, including fertilization. In fact, they were the first organisms that were used to understand conditional specification. In the case of sea urchins, fertilization is external. When we look at the life cycle of sea urchins, once a zygote is formed, the zygote undergoes embryogenesis to form a larva that is also called as a pluteus larva. That larva then undergoes metamorphosis to form a juvenile sea urchin that will then mature to form an adult sea urchin. Since fertilization in sea urchins is external, the male and female gametes are released in the water. The male gametes are able to find the female gametes or the eggs because the eggs secrete chemoattractants that are able to stimulate the male gametes, which then swim towards them through the process of chemotaxis. Once the male gamete is in the vicinity of the egg, the exocytosis of sperm acrosomal vesicle occurs. The enzymes in the acrosome are then able to create a path for the sperms so that they're able to bind to the vitelline envelope of the egg. Once they're able to pass through the extracellular matrix, the sperm can then bind to the egg cell membrane. And once this binding occurs, there is fusion of the egg and the sperm cell membranes. When the plasma membranes of the male and female gamete fuse, there is an increase in the calcium ion concentration in the egg cytoplasm, which results in the cortical granule reaction. The cortical granule reaction results in the formation of a structure called as the fertilization envelope. So when we look at the figure, we're able to see an egg that is surrounded by sperm. However, in panel B, when one sperm is able to fuse with the egg where the arrow is, we can see the formation of the fertilization envelope. The fertilization envelope covers the entire fertilized egg, and thus it is able to form a protective covering. The fertilization envelope protects the zygote as it undergoes further development. Let us now look at how cleavage occurs in sea urchins. So sea urchins show radial holoblastic cleavage. The first cleavage is meridional, which means the plane of cleavage is parallel to the animal and vegetal pole axis. This can be observed in the figure in the blue box, where we have the single-cell zygote and the plane of cleavage is shown, and it is parallel to that animal pole that is on the top and the vegetal pole that is at the bottom. So the axis joining the animal and vegetal pole is parallel to the plane of cleavage. So this gives rise to two cells that have all contents of the animal and vegetal pole. In the second cleavage, we still have a meridional cleavage, but the plane is perpendicular to the first plane of cleavage, and this is what is shown in the blue box. So by the end of the second cleavage, we now have 4 cells, and all 4 cells still have all information in terms of proteins and mRNA that is present in the animal as well as the vegetal pole. Now in the case of the third cleavage is a little different. The third cleavage is equatorial. So when we look at the figure or the part of the figure in the blue box, the plane of cleavage is perpendicular to the animal vegetal pole. And this causes a separation of the animal and vegetal hemispheres in the daughter cells that are formed. And that is what we can see in the figure in the blue box. Now, in the fourth cleavage, what happens is the animal tear cells, which is the part that is on the animal side, they divide
meridionally to form equal-sized daughter cells called as mesomeres. This is what is basically shown in the box. So in the box, when we look at the animal half of the cells, they have undergone meridional cleavage to form equal-sized daughter cells that are called mesomeres. The vegetal half, on the other hand, undergo equatorial plane of cell division, and however, when cell division occurs, it is unequal. So we actually end up with larger cells called macromeres and smaller cells called micromeres. In the fifth cleavage, as shown in the blue box, the mesomeres will divide equatorially to form two tiers. One is called as AN1 and the other is called as AN2. This is what is shown in the smaller blue box. Now what happens to the macromeres? So the macromeres that are below, they divide meridionally to form 8 cells, and their location is right below AN2. The micromeres don't divide immediately, but after a while they will also divide to form large micromeres and small micromeres. Now after the sixth cleavage, the cells can be assigned to different tiers. For example, when we look at the blue box, the cells right off the top were derived from the AN1 tier. The dark blue cells are the AN2 derived cells. Then we have the vegetal 1 derived cells and the vegetal 2 derived cells. It's not shown in this figure, but ultimately we will also have large macromeres and small micromeres. Okay? Let us now go to the next stage, which is the blastula formation stage. Now, as the cells undergo cleavage and more cells are formed, they start arranging themselves to form a central cavity, which is called as the blastocele. So when we look at the figure to the left, this is where we are seeing the cells undergoing cleavage. And when we look at the figure to the right, the cells have now gone to the periphery or to the edges of the embryo, and so in the middle we have a central cavity, which is the blastocele. Now the cells that are at the periphery, they associate with each other with tight junctions, and that's how they're kept together. These cells will then develop cilia, which are these small hair-like structures. The cells at the vegetal pole will start to thicken to form a vegetal plate. And in the figure to the right, we are actually seeing the thickening happening where you can see that yellow curved line. Once all of this happens, we have the blastula stage, and the embryo will digest the fertilization envelope that was protecting it and becomes a free-swimming hatched blastula. Now we should note that in this stage, we still have not had gastrulation. So once the hatching happens of the blastula, the gastrulation process can begin. Before going into the gastrulation process, let us look at the different fate maps that have been generated.
With this, we come to the end of our talk, where we learned about the life cycle of sea urchins, as well as the fertilization and cleavage processes that occur in sea urchins. We then learned about the progression to the blastula stage in sea urchins.